Hi guys, we're here to find out more about the phenomenon known as CRISPR. What is CRISPR, you ask? Well, luckily we have the knowledge of Dr. Cook Poo, a renowned scientist in microbiology with genetics. Here, let's come find out. Mr. Cook Poo, what have you been working on?
Oh, why hello there, Mr. Trump. I didn't see you there. What I'm working on right now is this thing called CRISPR. CRISPR is something that has allowed us to modify DNA with extraordinary accuracy, and we've been able to do things such as fake blindness by mutation and even inhibit hepatitis C.
Oh, really?
Yes. But what exactly is CRISPR? Well, let's find out.
CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. They are segments of prokaryotic DNA that contain short repetition of base sequences, which are the ones in the blue. These are base repeats. Each repetition is followed by short segments of spacer DNA. Spacers are simply regions of non-coding DNA. You know how not all DNA is used to code for certain parts of the body? Well, that's what spacers are.
CRISPR was first discovered in the DNA of bacteria in 1987 by scientists in Japan. It functions within the bacteria's immune system. How it works in bacteria is that when an invading source of DNA is encountered, that little guy right there, it then copies and incorporates segments of the foreign DNA into their genome as a spacer between the repeats. They enhance the bacteria's immune system's response by providing a template, or a guideline, for the RNA molecules to be able to identify and target the DNA sequence in case of future infections.
So, when the DNA enters the bacteria again, the RNA recognizes the incoming sequence and will guide the CRISPR complex to that. Once the CRISPR complex is at the sequence, Cas9, or CRISPR-associated proteins, will cut and splice the DNA and disable the invading gene. Snip DNA with unparalleled precision. In the fall of 2012, researchers from UC Berkeley found a way to use the CRISPR immune system to create a new gene editing tool. They could program the system to identify, cut, and even replace any gene sequence. With expert precision, of course. Now what exactly is Cas9? Well, Cas9 is the enzyme that snips the DNA. But what is CRISPR then? CRISPR is nothing more than just a collection of DNA sequences that tells Cas9 what to snip. So essentially, CRISPR is the blueprint, Cas9 is the worker. He reads the instructions and it goes and does its job. So it's actually very simple. Essentially, all that the scientists have to do is feed Cas9 the correct CRISPR sequence, and then have a guide RNA, and boom, you can cut and paste bits of DNA sequence into the genome wherever you want. Wow.
So what can CRISPR do? It can do a variety of things to the human body, like fix blindness by mutation. It can also inhibit hepatitis C, and even cure HIV, but possibly. Pretend this box is a genome. The sides are the repeats, and the red part in the middle is infected by the HIV virus. In the CRISPR system, we take a very precise pair of scissors called Cas9 and snip a part or even all of the HIV genome, then reattach the severed ends of the human genome.
So now that you cut out a piece of the genome, it's incomplete. Does that mean the HIV genome is disabled?
Indeed! If scientists could do that, the hope is that this would be a step on the road to getting a functional cure for HIV.
Wow. My hat sure does hurt from all that writing. Hope you learned a lot.
Yeah. Yeah, it was very important, Mr. Cook Poo. Yeah. We learn a lot. Thanks to you.
How would you like to give everyone a little recap?
Yeah, sure. Let's give them a recap, but let's head over to the whiteboard. Over here. Here. Over here.
Okay, so, to recap, are you ready?
Yup!
What does CRISPR exist in?
In bacteria's immune system.
Good job. Who discovered it and when?
The Japanese in 1987.
Wow. Good job.
Thank you.
Next, what were the scientists at UC able to do with CRISPR?
Edits and targets DNA with groundbreaking accuracy.
Okay, one more question. What does Cas9 stand for?
CRISPR associated proteins.
Wow, look at that. Cas. Just kidding, we have one more question for you. What does CRISPR stand for?
Clustered repeats?
No, it stands for clustered regularly.
Regularly. Interspaced, short, palindromic repeats.
Wow, good job. Do you know what you deserve? You deserve a high E.
A high E?
Wow, that was very informative, Mr. Cook Poo. Now, to end our series, we're gonna go to the washroom. Have all a good night and stay excellent. Oh, you're still here? No, go, please go. It's over. Documentary's over. Get out. Yo, put my name first. Put my name first.
Nice.
You even spelled my name wrong.